Welcome back to the channel. This is Ian.
And this is Undra. We're so glad you're here with us today as we explore the fascinating world of cancer's secret energy playbook, unpacking metabolic shifts in colorectal tumors.
That's right. It's going to be an interesting discussion. Okay, so let's unpack this challenge first. How do scientists even begin to study the really intricate stuff happening inside cancer cells?
Especially when they've only got tiny tissue samples, right? Like from a biopsy.
Exactly. We're talking just a few milligrams. It feels like trying to understand a whole factory by looking at, I don't know, one tiny gear.
That's a great way to put it. And yeah, the challenge is huge. Inside that tiny bit of tissue, you've got thousands of different molecules.
Metabolites, lipids, all the cell's working parts and fuel.
Right. And many are there in really, really small amounts. So you absolutely need tools that are incredibly sensitive.
And precise, I imagine, to tell everything apart.
Extremely precise. Tools that can identify and measure this huge variety of molecules all at once from that very limited starting material.
So let's dive into those tools then.
Yeah.
What are we talking about here? What allows researchers to look at these tiny, complex samples?
Well, one of the main workhorses for this kind of thing is liquid chromatography-mass spectrometry, usually just called LCMS.
LCMS, okay.
Think of it like a two-part process to analyze the sample's contents.
And the first part is the LC liquid chromatography.
Exactly. So imagine you have this mix of all the molecules from the cells. The LC part works like a sorting system.
Sorting them how?
Based on their chemical properties, like how they interact with a surface inside a column or how well they dissolve in the liquid moving through it. It basically separates the mixture out into different streams of molecules over time.
Okay, so it sorts the molecular jumble into neat lines. What's next?
Then those separated streams flow directly into the mass spectrometer, the MS part.
And that weighs them.
It does, but with incredible accuracy. It measures the mass-to-charge ratio of the molecules, and since each unique molecule has a unique mass.
Ah, that tells you what it is.
Precisely. And it also tells you how much of it was in that stream, which relates back to how much was in your original tiny sample.
Okay, so separate them, then weigh them to identify and quantify. Sounds straightforward in concept.
But incredibly challenging in practice, especially with just milligrams of tissue.
Yeah. So standard LCMS might not be sensitive enough.
Often not for these really small, precious samples. And that's why the research we were looking at used a specialized version, capillary scale UPLCMS, or CAP-LCMS.
Capillary scale. What does that mean?
It means everything is miniaturized. They use extremely thin columns like capillaries and much, much lower flow rates for the liquid.
And making it smaller makes it
more sensitive. Dramatically more sensitive. It concentrates the sample, allowing the mass spec to detect molecules even at very low concentrations.
Which is exactly what you need for a tiny biopsy.
Absolutely essential for getting good data from maybe just 3 to 5 milligrams of tissue, like in this study.
Wow. Okay, so this super sensitive cap LCMS is key.
It is. And, to get a really broad view of all the different types of molecules.
Because not all molecules are the same, chemically speaking.
Exactly. Some like water, some hate it. Some are fats, some are sugars. So this study actually used 2 different CAP-LCMS methods, complementary ones.
2 methods on the same sample. Why?
To cast a wider net. One method used something called HILIC, that's hydrophilic interaction chromatography.
Hydrophilic. Water-loving.
Right. So HILIC is good for separating polar molecules, things like sugars, amino acids, those sorts of water-soluble compounds.
Okay, HILIC for the water-friendly stuff. What was the other one?
The other used reversed phase chromatography, or RP, and that's sort of the opposite. It's better for non-polar molecules.
Like fats and oils.
Exactly. Lipids, fatty acids, things that don't mix well with water. So by using both HILIC and RP, CAP-LCMS,
they could see almost everything, both the water-soluble and the fatty molecules.
They got much closer to seeing almost everything, yes. It gave them incredible coverage. The study managed to annotate, meaning identify and measure, about half of all known human metabolites.
Half? From a few milligrams. That's amazing.
It really is. And around 700 different lipids, too. That level of detail and sensitivity from such small samples is pretty powerful.
Was it fast? Could they analyze many samples?
Relatively fast, yeah. The run times were quite short, like 14 or 20 minutes per sample, which allows for what researchers call high throughput, analyzing lots of samples efficiently.
Okay, that's fascinating on the tech side. So how did using these really advanced tools help them understand colorectal cancer specifically?
Right, this is where the biology gets interesting. They applied these LCMS methods to analyze human tissue biopsies.
From different stages of colorectal cancer.
Exactly. They had samples of normal, healthy colon tissue. Then they had samples of precancerous polyps. The technical term is adenomas.
Okay, the stage before it becomes cancer.
Yes. And finally, samples of actual colorectal cancer tissue, the carcinoma.
So they could track the changes across these stages. What were they looking for?
They were looking for metabolic alterations or metabolic reprogramming. It's this idea that cancer cells fundamentally change how they handle energy and nutrients.
They rewrite their internal energy playbook to help them grow.
That's a perfect way to put it. They change their metabolism, how they break down sugar, how they use amino acids, how they make building blocks to support their rapid growth and survival. This altered metabolism is now seen as a key hallmark of cancer.
So they're hunting for these metabolic differences. Did they just look at the metabolites?
Not just metabolites and lipids using metabolomics and lipidomics, they were clever and also integrated transcriptomics data.
Which looks at gene activity.
Right. It measures which genes are being turned on or off, so they could see if changes in the molecules present were linked to changes in the genes controlling the metabolic pathways. It gives you a much fuller picture.
And did they find clear differences? Could they tell the tissues apart just by their metabolic profile?
Oh, absolutely. The analysis showed that the tissue type normal, polyp, or cancer was the most dominant discriminating factor, meaning the metabolic pattern alone was a very strong indicator of whether the sample was healthy, precancerous, or cancerous, like a metabolic fingerprint for each stage.
Okay, so a clear fingerprint. But here's a question that comes to mind. Does this big metabolic shift, this reprogramming, only really happen when it becomes full-blown cancer?
Or does it start earlier?
Yeah, does it kick in during that polyp stage?
And this is where it gets really interesting. One of the major findings of the study was that actually many of these significant metabolic changes are already happening in the precancerous polyps, the adenomas.
Wow. So it's not just a switch that flips when it becomes cancer.
No, the metabolic groundwork is being laid much earlier. It starts in the polyp stage.
Okay, that's a huge insight. So the secret energy playbook is being drafted even before it's truly cancer. What kinds of changes did they see happening early on?
They pinpointed specific metabolic pathways that were altered in a similar way in both the polyps and the cancer tissue compared to the normal tissue.
Which pathways?
Well, think about core processes. Glycolysis, for example, that fundamental pathway for breaking down sugar to get energy. That was already changing in the polyps.
Altering energy production early. What else?
Nucleotide biosynthesis. That's the process cells use to make the building blocks for DNA and RNA.
Which they need a lot of if they're starting to divide more, right?
Exactly. And that pathway was upregulated, more active in both polyps and cancer. Also, the way cells use certain amino acids building blocks like serine, glutamine, proline, that metabolism was shifting early too.
So fundamental processes for energy, for building new DNA, for using amino acids already different in polyps. Did the gene activity data back this up?
It did. The transcriptomics showed that the genes coding for the enzymes that carry out these reactions, like PSAT1 involved in making serine, or genes for nucleotide synthesis, these genes were often more active, upregulated in both the polyps and the cancer.
Okay, so something is driving these changes, even before it's technically cancer. Any ideas what that might be?
Well, the researchers pointed towards a potential key player, the MYC gene.
MYC, I've heard of that one. It's involved in a lot of cancers, isn't it?
It is. MYC is a transcription factor. Think of it like a master switch that controls hundreds, maybe thousands of other genes.
Okay.
And it's known that MYC often gets turned up, upregulated very early in colorectal cancer development. What they found was a strong correlation
Between MYC and, yeah.
between the activity of MYC itself and the genes it controls, and the activity of those specific genes involved in glycolysis, nucleotide synthesis, amino acid metabolism. The pathways changing early.
So it strongly suggests that MYC might be the conductor leading this metabolic orchestra, starting the changes in the polyps.
That's the hypothesis, yes. It seems like MYC could be orchestrating these early shifts needed for increased growth, even at the precancerous stage.
Okay, that makes a lot of sense. So if many of the types of changes start early in the polyp stage, what's different when it actually progresses to full cancer? What ramps up the aggression?
That's a really important distinction. While the kind of metabolic rewiring often begins in the polyps, the magnitude or the intensity of those changes frequently becomes much greater in the cancer.
How so?
Many specific metabolites showed much bigger changes, what scientists call higher fold changes in cancer compared to polyps, relative to normal tissue.
So more dramatically increased or decreased?
Exactly. For instance, things like glutamate, glutamine, even glucose concentrations. Their increases were often significantly more pronounced in the cancer samples than in the polyp samples.
So the Playbook's themes are set in the polyp stage, but in cancer, it's like they turn the volume way, way up on those specific metabolic pathways.
That's a good way to think about it. And they found another key difference specifically in the cancer tissue. It involved something called solute carrier genes, or SLC genes.
SLC genes, what do they do?
These genes provide the instructions for making transporter proteins. These proteins sit in the cell membrane.
The outer wall of the cell.
Right, and they act like specific doorways or pumps, controlling what gets into and out of the cell, nutrients, waste products, ions.
Like specialized gates for importing supplies.
Precisely. And here's the crucial finding. Certain SLC transporter genes were found to be significantly overexpressed, meaning much more active, only in the cancer samples, not in the polyps.
Exclusively in cancer. Which ones?
They highlighted transporters for key nutrients that rapidly growing cancer cells desperately need. For example, transporters for glucose, GLUT1, was mentioned as a key one.
Bringing in more sugar fuel.
Yes, and also transporters for amino acids like glutamine and glutamate. They mentioned LAT1 as an example there.
This is like the cancer cells install these extra high-capacity VIP doors just for the fuel they crave the most, something the polyps hadn't done yet.
That's a perfect analogy. This increased ability to actively suck in essential nutrients likely gives the cancer cells the extra edge, the extra fuel and building blocks they need for their much faster, more aggressive growth compared to the polyps.
That makes sense, fueling the fire.
And interestingly, they noted that some of these transporters, like GLUT1 and LAT1, might even be useful as predictive markers, potentially indicating how a patient might respond to certain treatments.
Fascinating. Okay, so we've talked sugars and amino acids, but the study also looked at fats, right? The lipidome. What did they find there?
Yes, the lipid changes were also quite significant, though maybe a bit more varied between samples. But one really striking finding involved triglycerides.
Triglycerides. Those are the main way we store fat for energy, right?
That's right. And what they found was that triglycerides containing specific types of fatty acids, very long-chain polyunsaturated fatty acids, or VLC-PUFAs.
That's a mouthful. VLC-PUFAs.
It is. These VLC-PUFA-containing triglycerides were significantly increased in both the polyps and the cancer tissue compared to normal.
Increased early on, in the polyps too.
Yes. They mentioned specific examples like arachidonic acid and eicosapentaenoic acid being incorporated into these fats, showing up as early lipid changes.
So specific long unsaturated fatty acids are building up. Was anything decreasing?
Yes, on the flip side, triglycerides containing short saturated fatty acids were found to be decreased.
So the cells aren't just storing more fat overall, they're actively changing the type of fat they're storing or making.
Exactly. It suggests a shift in the composition of the fats, and they looked at the genes involved too.
To see if the cells were making these fats themselves.
Right, and they found increased activity, higher gene expression for enzymes involved in building fatty acids and also elongating them, making them longer.
Which supports the idea that the cells are actively synthesizing these specific VLC-PUFAs from scratch, what's called de novo synthesis.
Precisely. It looks like they're deliberately building these specific types of fats.
Why would they do that? Are these fats just for energy storage?
Probably not just for energy. These specific VLC-PUFAs, like arachidonic acid, aren't just inert storage molecules. They can be used to make signaling molecules.
Signaling molecules, like messengers.
Yes, messengers involved in processes like cell growth and inflammation. The study mentioned the COX-2 pathway, which uses arachidonic acid, and is known to be important in colorectal cancer development.
Ah, so these altered fats might also be feeding into the signaling pathways that drive the cancer's growth and spread.
That's a strong possibility. So the lipid changes aren't just about fuel, they could be actively contributing to the cancer's biology.
Wow. Okay, this has been an incredibly detailed look under the hood, seeing how colorectal cancer rewrites its energy playbook, starting even in the polyp stage, and then really ramping things up in full cancer, all revealed by these super sensitive analytical tools.
It really paints a picture of cancer's metabolism not as chaotic, but as a systematic evolving process. Changes start early, maybe driven by master genes like MYC.
And then become more aggressive, fueled by installing those specific nutrient transporters.
Exactly. And it's amazing the level of detail they can get from just those tiny biopsy samples now. Things like specialized software, like the cosmic software mentioned by one of the authors, are really key to digging out these subtle signals from complex data.
So, stepping back, why is this important for you listening at home to know about? Why does understanding cancer's metabolic playbook matter so much?
Well, getting this deep understanding of the specific changes, what fuel cancer uses, how it acquires it, how this changes during development, it's incredibly valuable.
For scientists and doctors.
Yes, it potentially opens up completely new strategies.
Such as?
For one, think about early detection. If we know these specific metabolic markers, these fingerprints, are present in precancerous polyps.
Maybe we can develop tests to find them, catch things much earlier.
That's the hope. Catching it before it even becomes invasive cancer would be huge. And secondly, it points towards new targeted therapies.
How so?
If we know cancer cells are uniquely dependent on, say, bringing in glucose through that GLUT1 transporter, or reliant on making those specific VLC-PUFA fats,
maybe we can design drugs to block just those processes.
exactly. Develop therapies that specifically disrupt the cancer's unique way of fueling itself or building itself, potentially with fewer side effects than traditional chemo that hits all dividing cells. It's about finding the cancer's metabolic Achilles heel.
It really highlights how these incredibly detailed investigations at the molecular level, understanding the nuts and bolts inside the cell, can genuinely open up new avenues in how we fight cancer. This has been a truly fascinating exploration into the hidden energy secrets of cancer cells.
It really has. There's just so much intricate biology going on, and studies like this really help connect the dots, between the genetic changes we hear about and what's actually happening metabolically inside the cell.
Thanks so much for joining us for this exploration.
We hope you learned something new and interesting today.
We'll see you next time for another deep dive.
Bye, everyone.
See ya.